Hello, everybody. Welcome to today's lecture, and we continue covering the cellular level of organization. Now, I mostly cover this chapter, so if you want to watch the first part of this PowerPoint, then please watch the cellular level of organization part 1 video. Right, so. And today we're gonna start with physiology part, right? So let's go ahead and begin. Now, in this chapter, we are talking about cells. And if you remember, we began our conversation covering cell theory. And cell theory states that cells are structural and functional units of life. Cell is the smallest unit of life, and all cells come from pre-existing cells, right? So how everything begins. Now, any organism begins with one cell, right? That first cell is called zygote. Now, when secondary oocyte is fertilized by sperm, it became ovum, and then zygote is formed. Now, zygote carries all DNA that you need to build your body, right? Zygote covers DNA, half of genetic material from your mother and half genetic material from your father, right? Now, this first cell, zygote, is called totipotent cell. It's a stem cell. Stem cell means that a cell can self-renew. So it will divide and form more stem cells, right? And totipotent means this cell have potency to differentiate in every single cell and tissue in your body, right? So zygote will split into two cells. Those two cells will split into 4 cells, 8, 16, 32, until we form a ball of cells. And all those cells are still totipotent. So their destiny is to form every single tissue and every single organ in your body, right? Then time passes, and this ball of cells that is called morula will start differentiating. So now cells are not identical. They start forming our first embryonic tissues, right? And some cell will form endoderm stem cell progenitors. Some will form mesoderm stem cells progenitors, and some ectoderm. Now, those cells now, they are not totipotent anymore. They are pluripotent. They still can differentiate in many tissues and organs, but not to any tissue and organ, right? So if you look over here, endoderm line cells, they can differentiate into your lungs, pancreas, liver, GI tract, right? But they cannot now become your muscles or your blood cells or your skin or your neurons, right? So still lots of stuff will happen with those cells. They will differentiate further and further, right? But right now they are pluripotent, right? That makes sense. So those are pluripotent. So they stem cells, so they can renew themselves, and they will form your organs and tissues. Now, when cells continue to differentiate, so they become different, they form different lines and lineages, they now, instead of pluripotent, became multipotent. Now, multipotent, those are cells that found in adults, right? So it says over here, multipotent adult bone marrow, skin, some cells that will form your blood, right? Now, these multipotent cells further differentiate into unipotent progenitor cells, and unipotent cells, they still differentiate, but they differentiate in one lineage of cells only, right? So to sum up what we just said, potency of a stem cell or potency of progenitor stem cells will show you it will show you how many tissues and organs can be formed for that from that particular family of stem cells, right? Now this arrow over here actually very important, and this is a new research that was done not long time ago, and what they find out, if we have cells that already, let's say, multipotent or oligopotent, or even cells that already differentiated into adult cells, like fibroblast cells, for example, we can induce them and reverse their development back to stem cells. So we can, like before, to get those pluripotent cells, we needed embryo, right? And when you need embryo to harvest cells, then lots of issues arise, and you know, is it ethical or non-ethical to grow embryos that potentially now can be a human being, and now you harvest their cells. But now with this new research that was done in Japan, and I believe even they get a Nobel Prize for it, what they find out that you can use cells from adult, right, like fibroblast, you can add some chemicals, right, some transcription factors, and you can make them, you reprogram them back to stem cells, right? So that's what induced pluripotent stem cells are, right? So very promising research, very interesting to kind of keep an eye what's going to happen with this stem cell induced pluripotent stem cell research. Okay. Now, this is example of cell differentiation. So when we, let me exit for a second. We're gonna start with multipotent stem cell, hemocytoblast, and this diagram show you, and now this, first of all, this is adult stem cell, so it found in a red bone marrow. So this cell, I'm go back, it's totipotent cells. Oh, I'm sorry, it's a pluripotent cell. Oh, no. Oh my goodness. Okay, guys. Okay, so now why it cannot be totipotent or pluripotent? It cannot be totipotent or pluripotent because those are embryonic cells. Now we are talking about development of blood cells in adult, right? Not when it, you know, it happened, not that time when you're still embryo. Now we're talking about embryonic, or not embryonic, but development of blood cells in an adult organism, right? Keep it in mind, adult, we're talking about adult. So if you're talking about adult, it cannot be totipotent, it cannot be pluripotent, because those are embryonic stem cells. So it can be only multipotent cells, okay? So good, I apologize for that. So multipotent stem cells, they found in your red bone marrow, they will differentiate into myeloid progenitor cells and lymphoid progenitor cells. Now, after they commit to their future destiny, after they differentiate in these two progenitor cells, now instead of multipotent, they became oligopotent cells, right? That makes sense. That's why I'm constantly constantly going back to the slide to show you. We're going to start with multipotent, because in this example, we cover adult hematopoiesis, right? And those hematopoietic stem cells, multipotent, became oligopotent myeloid progenitor and lymphoid progenitor cell. Now, myeloid progenitor cells, they will differentiate into thrombocytes that are your platelets, erythrocytes, red blood cells, mast cells, those cells that are important in inflammatory process, and many white blood cells. So all cells over here are white blood cells: basophils, neutrophils, eosinophils, and monocytes. Now, monocytes, they are monocytes when they're still in the bone marrow. Now, when they start moving to tissues, they will differentiate into macrophages and dendritic cells. So most of the cells found in your blood are cells that form from myeloid progenitor cells. That makes sense? Now, lymphoid progenitor cells, they will differentiate into natural killer cells and one of the smallest cells in your body, right? The smallest cell is erythrocytes. So erythrocytes and sperm cells are two smallest cells, and lymphocyte is pretty small as well. Now, small lymphocyte, and this lymphocyte further will mature into T lymphocytes and B lymphocytes, and those are important in your immunity, right? They're all important in your immunity, but you have now T lymphocytes and B lymphocytes are important in type of immunity that we call adaptive immunity, because you have innate immunity when pretty much you fight any pathogens, you don't develop any memory against antigen, right? So innate immunity, you are born with that immunity, and basophil, neutrophil, eosinophils, macrophages, dendritic cells, natural killer cells, they are all part of your innate immunity. Now, your adaptive immunity that you develop through your life, those cells that are important for adaptive immunity are T lymphocytes and B lymphocytes. B lymphocytes then mature into plasma cells, and plasma cells, the ones that will produce antibodies to inactivate foreign pathogens like bacteria or viruses. Right, so this is an example of cell differentiation that happen in adults in a red bone marrow, right? And this is how all blood cells are formed. Okay, now for some reason, I cannot move to the next slide, so I need to stop it for a second. Okay, so I'm back. I don't know what's going on with my Zoom, but for some reason, once in a while, I cannot move to my next slides when I'm in the presentation mode, right? Let's see. So here's, yeah, now it's working. Now, not only cells are formed, right? They form from that first zygote, right? Early at very, at the very beginning of your development. Cells differentiate, but sometimes cells need to destroy themselves, right? For example, you might have old and non-working, non-functional organelles, cytoplasmic beads that are accumulating inside your cells, and you want a process that would allow you to get rid of of all these non-functional organelles. And this process is called autophagy or self-eating. Phagy means eating and auto means your cells pretty much eating themselves. It's a process of disposing of non-functional organelles, cytoplasmic bits, by forming autophagosomes, which can then be degraded by lysosomes. And this diagram show you process briefly, right? For example, you have mitochondria that is non-functional. Now the phaga form, or phagosome will be formed. You see how they progresses pretty much, but we just can call it phagosomes. So that vesicle will be formed from endoplasmic reticulum surrounding this non-functional organelle. Now this autophagosome then fuse with the lysosome. And if you remember, lysosomes carry digestive enzymes. So those digestive enzymes will be released and they will break down this mitochondrion, right? And your cell can recycle all the organic molecules that was made over here from a large parts of mitochondria, or your cells might just get rid of it through the process called exocytosis, right? So cells do have a mechanism that allows them to get rid of non-functional organelles, autophagy. Well, sometimes cells will go through apoptosis. It's a programmed cell death that causes certain cells to neatly self-destruct. Some apoptosis happen even during embryonic development, right? When you form your body, you might want to get rid of some cells. But apoptosis might happen with cancer cells, infected cells, old cells, and it's a very beneficial process for an organism, because if you have a cancer cell and you don't want it to divide, because when cancer cells divide, they form tumors, and those tumors can metastasize. So it's better for those cells to self-destroy, right? Go through apoptosis. Apoptosis involves mitochondria. It begins with mitochondrial membranes leaking chemicals that activate enzymes called caspases. Caspases cause degradation of DNA in cytoskeleton, which leads to cell death. Dead cells shrink and phagocytized by macrophages. Right now, on this diagram, what is interesting about this diagram, it shows you relationship between autophagy and apoptosis. So sometimes they can act independently, and both of them can cause cell death. But remember, this is the beneficial process. This is a kind of programmed process that allow you to clean your body of the non-functioning cells. Or they can be self-induced, so one can lead to the other, like autophagy can lead to apoptosis, or apoptosis will lead to autophagy, and overall result will be cell death. Or sometimes autophagy antagonizes apoptosis, right? So instead of, let's say, killing the cell, you can just remove whatever non-functional organelles you don't need inside, right? Okay. So over here we can see images of cells, and its electron microscope is used. This is a normal cell. This is cell that goes through apoptosis. This cell goes through autophagy, and this is necrosis. Now, necrosis is death of the cell, and it's not programmed and it's not planned. So necrosis might happen because of inflammation, or lack of oxygen to your cells, right? Or kind of like physical damage of the cells. And this is a process that usually, necrosis always has inflammation involved. Now, what else? See what else is different. So general morphology. When we have apoptosis, cells shrink. When we have necrosis, cells swell. Chromatin condensation, we have high degree of chromosome condensation through apoptosis. We have some condensation when we have autophagy and no condensation during necrosis. Caspase activation happen only when we have apoptosis, right? So this is the way your cells die, right? So if you wonder why we're talking about this stuff, right? Because this chapter is about your cells. And go back to the first PowerPoint and okay, I don't know. Okay, so it doesn't say over here, but yeah, cells come from pre-existing cells. It says right here, sorry, cells can arise only from other pre-existing cells, right? That's why we are talking about differentiation and stem cells, how all your cells came from this first zygote, right? And we cover briefly example of cell differentiation, and our example was hematopoiesis, or formation of blood cells, all blood cells, red blood cells and white blood cells and platelets, right? And because cells are formed, sometimes cells need to die. And this, we have 3 processes of cell death, right? 2 of them are programmed and controlled, and 1 is not, necrosis, right? Okay, and there is one more topic we want to cover in this chapter, and it's called cell membrane potential. Now, before talking about cell membrane potential, let's just look at something that is very familiar, a battery, right? If you have a battery, you know that your battery has voltage, right? Now, what is voltage? Voltage is electrical potential energy. You know battery has energy, right? Because battery, they can do some work. For example, if you have a battery, your light bulb can work, or your computer works, or your remote control is working, right? Because of your watch. So batteries store this potential energy that we call voltage. And voltage is separation of opposite electrical charges. So on a battery, you have a positive end, negative end. So the charges are separated. And when you connect these negative and positive ends, you will get electrical current. And this is also energy, right? That this is kinetic energy now, because electrons are moving through this wire, and they can make some devices work, right? Like your light bulb, or your watch, or your computer, or your phone, and so on, right? So the point is, if you separate charges, right, you can store energy, and then you can use this energy to do something, right? Now, your cells, every cell in your body does this. Your cells separate charges, right? They store this potential energy, and then they use this energy if they need to perform some functions, right? And when your cells separate charges, we call it membrane potential. So all cells establish electrical potential, right? This, right? Called the resting membrane, cell membrane potential. And it can be measured. So if you measure it, it will be minus 50 millivolts to minus 100 millivolts. And it has this minus side to show you that inside a cell, the charge is negative, right? Okay, so let's look over here. So imagine this is your cell, right? And you have negative charge inside, you have positive charge outside. So they start looking like a battery, right? And if you use voltmeter, so that's a device that measures voltage, you can measure this amount of volts. And it will show you like here, -70 to -100 millivolts. Now, how much it is? Is it a lot? Is it a little? Here, compared to batteries that you know, so you have like AA, AAA, CD batteries. They are 1.5 volts, right? Now, let's just imagine that this is 100 millivolts, right? Just to make our calculation easy. 100 millivolts then equals 0.1 volts. So, and those batteries are huge, right? Your cells are microscopic. So every cell in your body store 0.1 volt of, so it established this membrane potential, and voltage is 0.1. So it's huge, right? For these tiny cells. Now, if you add all the cells, those are, you know, millions of cells that make up your body, and each one has this voltage, you can imagine the amount of electricity in your body is amazing, right? Now, so pretty much here we reviewing what I just told you one more time. Separation of oppositely charged particles or ions across a membrane creates membrane potential. Potential energy measured as voltage. Resting membrane potential, RMP, is a voltage measured in the resting state in all cells. Many students think that a resting membrane potential only exists in neurons and muscle cells, but no, it's actually in all cells. And again, they show you pretty much the same. You have cell, and if you measure voltage, so this is the difference between what you have inside and outside, that's your resting membrane potential, right? Make sense? Okay, well now, interesting part, how is it formed? Because it's a physiology, right? In anatomy, it would be just enough to say your cells separate charges, and separation of charges is called voltage, and you can measure this, and it will be like -70 millivolts, and your cells can use this potential energy to do some cellular processes. That's enough for anatomy. It's not enough for physiology, because in physiology, we need to understand how exactly cells are able to do it, right? Now, I will try to make it really simple without any, you know, small details. Now, look over here, what you have. You are shown a cell, right? Imagine it's a cell. So this kind of yellow part is cytoplasm. Blue part is plasma membrane or cell membrane, and this is extracellular fluid, right? So this is outside a cell. Now, inside a cell, you have proteins. Proteins are pretty big molecules, and they cannot leave your cells. They stay inside, right? Because they're too huge to move through cell membrane or even through channels. Proteins have negative charge, right? So here, proteins shown that they are negative. So keep this in mind. Next, we know that in the plasma membrane, we have embedded proteins, transmembrane proteins. Some proteins are channels. So this is a channel protein. I want to remind you, channel protein means stuff can move through this channel in and out of a cell. So here's another channel protein. This is another protein inside plasma membrane, and this one is pump. It's sodium-potassium pump. This protein uses energy of ATP, and what it does, it constantly moves sodium out, potassium in, right? And we have for every pump, I would say for every action, for every exchange, 3 sodium out, 2 potassium in. right? And now think about it for a minute. If you constantly pumping sodium out, then you will have lots of sodium outside. If you constantly bringing potassium in, you will have lots of potassium inside, right? So your cells, so the most common ion, cation inside a cell is potassium. The most common ion outside a cell in extracellular fluid is sodium. In biology, we even have these sayings, cells are salty bananas. Salty, so if you imagine you have banana and you put salt on the top of banana, that's what in extracellular fluid. You have sodium and chloride, by the way, and chloride ions. And you know, banana is rich in potassium, so you have potassium inside, right? So because of sodium potassium pump, your cells create concentration gradient. Lots of sodium out, lots of potassium in, right? Now, so far, we have negative charge inside, and we exchange sodium for potassium, but both of them have positive charge, right? So it's not that big of a difference. So cells still should stay neutral, but we need to explain why cells are negative inside. Here, positive charges goes out, positive charges goes in. So exchange kind of like, well, not a big deal. It does make a little bit difference because 3 goes out, 2 only goes in, right? But it only will give you like -5 millivolts, not -70, not -100 millivolts, right? Now, something else needs to happen. Now, think about it. If you have lots of potassium inside, and you have a channel for potassium to move out, do you think potassium will move out? Yeah, because molecules and ions and, you know, any particles, they will move from high concentration to low concentration, right? So potassium is moving out. A sodium, if you have a channel for sodium to move in, right? And we have lots of sodium outside, you think sodium wants to move back in? Yeah, it does. However, if you read over here, plasma membranes are 50 to 75 times more permeable to potassium than they permeable to sodium. Now, this is what I want to do, because this membrane is about, 75 times less permeable to sodium. Let's just ignore this for a second, right? Let me draw. Let's just ignore this channel. Now, why we can ignore this channel for a minute? Because we have way less sodium that moves in than potassium moves out, right? 50 to 75 times. That's a big difference, right? Now, so again, we're pushing sodium out, potassium in, sodium out, potassium in, but we have exchange between positive charges. So positive charges goes out, positive charges goes in. So overall, the cell is inside is still neutral, right? You have positive charges right? So here you have positive charges and you have negative charges. So you pretty much stays like no charge, neutral, right? But because potassium moving out, look, potassium will move out. Every time now positive charge moves out, right? If positive charge is moving out and let's say 75 potassium moves out and only 1 sodium moves in, right? Because it's right there. It's more permeable to potassium. So if 75 potassium moves out and only 1 sodium moves in, well, now it's not an equal exchange. Now we're losing positive charges, and we're losing positive charges, the cell inside become negative. That makes sense? Okay, So this is how your cells separate charges. Now, sodium-potassium pump creates chemical gradient for sodium and potassium. Then potassium start moving out, taking positive charges out, and creates electrical gradient. This channel is called leakage or leak channel. And chemical and electrical gradient form electrochemical gradient that allow your cells to create resting membrane potential, right? Well, you let me know if this explanation makes sense. But what I'm trying to tell you, I'm trying to explain you why your cells are able to separate negative charges from positive. And negative charges will be inside, positive outside. And this is because of two important proteins, sodium-potassium pump and leak channels. Sodium-potassium pump makes chemical gradient, lots of sodium out, lots of potassium in. And now sodium and potassium start moving down its concentration gradient. But every time we're losing about 75 potassium, we only gaining 1 sodium. Now, if you're losing so many positive charges, right, you become negative inside. And this is what we call resting membrane potential, because your cells became a tiny microscopic battery with separated charges, negative inside, positive outside. Now your cell stored potential energy that we can measure in voltage, and your cells can use this energy to do its cellular job, right? Okay, so that's it for this chapter. I hope you guys enjoyed and it was helpful. Thank you for watching.